So in this lesson, we are going to extend what you have learned from your AS, which is mutation can cause a change in the protein coded by the gene. And when there's a change in the protein, it may also lead to change in the phenotype. So today what you learn is what is the effects of mutant allele. That means when there's a mutation leading to a new allele, which is called the mutant allele. So the effects of mutant allele on the phenotype. So what are the phenotype you're going to learn are the common genetic disease in human population. So we've got albinism, sickle cell anemia, haemophilia, and Huntington's disease. All these are due to the mutant allele. So the learning outcome says that candidates should be able to explain the relationship between genes and protein, which is an enzyme coded by the gene, and how this enzyme will actually affect the phenotype with respect to, so there are 4 examples here. Okay. So TYR gene, which code for tyrosinase, which code for tyrosinase, when it's mutated, it gives rise to albinism. HBB gene, which code for haemoglobin, when it's mutated, it gives rise to sickle cell anemia. FB gene, F8 gene code for factor 8, which is a blood clotting factor. When it's mutated, it may give rise to haemophilia. And HTT gene code for Huntingtin protein. HTT stands for Huntingtin, so it's Huntingtin, okay, leading to Huntington's disease if there's mutation. So this is very interesting. So we're going to look at this one. So of course, there are a lot of, this is a recap of all you've learned about the mutation. So this is in your AS slides. You can read this on your own. I will go through very quickly because you've learned it, okay? So when you talk about mutation, there are 2 types of mutation, either mutation of the gene or mutation of the chromosome. So when you say about mutation of the gene, that means there's change in the base sequences in the gene. When you talk about mutation in the chromosome, it's either change in the number of chromosomes or change in the structure of chromosome, whether structure becomes longer or shorter. Okay, so this is called mutation. So when a cell has got mutated gene, it's called mutated cell. And then the organism is called mutated organism. Okay. So this one is mutation can occur in any cell in our body. It can be in somatic cells or germ cells. So if it occurs in somatic cells, it will not be passed to the offspring. But if it occurs in germ cell, because germ cell can undergo meiosis to form gametes, then it can be passed to the offsprings. So that's how a parent that carry a genetic defect can pass it to the offspring through the gametes. Okay. And mutation is always considered as random and spontaneous. That means to say that it's by chance. How to think of it as by chance is, let's say you eat, right? Every day you eat. Once in a blue moon you eat and you bite your own tongue. So this is called spontaneous. Okay, you do not know when, and it's randomly happening. So mutation occur in this manner. It's random and spontaneous, means by chance. Okay, so this is the most important part. What happens when the DNA mutate? So when the DNA mutate, of course, the basic is that from DNA in a gene expression, the gene in the DNA will be transcribed into mRNA and translated to polypeptide, and the polypeptide will bend and fall into the protein structure, tertiary structure. And this protein will carry out its function. So a lot of time they are enzymes, right? Enzymes function based on the structure, especially active site. Then of course, you've got carrier protein, you've got receptor, you've got antigen, also can be protein. Okay, for today's, mainly we are looking at the protein. Enzyme is an example of protein. Okay, so once there's a mutation, there's a change in base sequence in DNA, therefore there's a change in base sequence in mRNA, there's a possible change in amino acid sequence in the polypeptide, and therefore possible change in protein secondary tertiary structure. So the enzyme cannot function normally, and that will lead to a change in the phenotype, which we're going to look at afterwards. Okay. So we're going to look at the 4 examples I've mentioned earlier, how mutation may affect the phenotype. So mutation of a gene coding for certain enzymes leading to albinism, sickle cell anemia, haemophilia, and Huntington's disease. Okay. So the following genetic diseases, which is also a type of phenotype. Okay, this disease is also a type of phenotype because you can see it in the person, are due to mutation. So they are called genetic disease because it's due to mutation in the gene. So genetic disease is an illness, is a disorder, is a malfunction of the body due to either chromosomal or gene mutation. So it's called genetic disease. So mutation can be chromosomal, mutation can be on the gene. So it can be, genetic disease can be due to the mutation in chromosome or gene. Okay, so these 4 types of phenotype due to mutation that you're going to learn. Albinism and sickle cell anemia is due to autosomal recessive allele. Okay? Huntington disease is autosomal dominant disease. Haemophilia is sex-linked recessive disease. Okay, so let's look at albinism first. So when you talk about albinism, means albumin means egg white, isn't it? Albumin means white color, right? No color, white color. So the feature that you can see is like the skin is fairer than normal, and sometimes even the eyes is red color or pale color or gray color. Okay, so albinism is a group of genetic condition that causes lack of dark pigment melanin. Okay, so our skin color is due to melanin. So totally or partially missing from the eyes, skin, and hair. Even the hair also, okay, has no color. Most types of albinism is an autosomal recessive. And individuals that are homozygous recessive will show albinism. So therefore, albinism is not so common because it is recessive. So you need the 2 recessive allele to be there in order to show the phenotype. So it's not so common because it's not easy to get 2 recessive allele to come together. Okay. So the occurrence is only 1 in 17,000 births worldwide. We have got many different types of albinism. So a different type of albinism affects the eyes and not the skin. This type is sex-linked, so there are many types. Okay. So due to the total lack of the pigment melanin, albinos have characteristics like pale blue or pale pink iris in the eye. So you can see that this picture here, this is pale blue iris, this is iris, and this is pale pink. Okay, and then the skin is very pale, the hair also very pale, everything, because no melanin, which is the pigment. Okay, so in some cases there is also vision problem, so they must avoid UV light, UV radiation, and may require to wear tinted glasses to assist with photophobia, means scared of light. Okay, so what is the cause of the albinism? It's because of mutation in the TYR gene. That means the gene that codes for the enzyme tyrosinase. So TYR stands for tyrosinase. Okay, so you look at this a reaction. You start with L-tyroxine, and it needs 2 tyrosinase enzymes in the presence of oxygen. First, from gene 1 that code for tyrosinase, have to convert thyroxine to DOPA. From gene 2, which code for tyrosinase, I'll say tyrosinase 2, it will convert DOPA to dopaquinone, and from there, this dopaquinone is needed to produce eumelanin and phenylalanine. So these 2 are melanin. So in order to produce melanin, which is the color pigment of our skin, eye, and hair, we need these 2 enzymes, tyrosinase, which are coded by these 2 genes. So when there's a mutation in this gene, gene 1 or gene 2, this tyrosinase will change shape. So when it changes shape, of course, the substrate cannot stick to the active site of enzyme, so DOPA cannot be produced, or DOPA cannot stick to the active site of this tyrosinase. Dopaquinone cannot be produced, melanin cannot be produced. So this is how the mutation in the gene 1 or gene 2 will give rise to albinism. So it's due to mutation in thyroxine gene. Okay. So tyrosinase, so this reaction you need to know. You need to memorize this reaction. Okay, not the structure, just the reaction. Tyrosinase is responsible for the melanin production in melanocytes. Cytes means cell. Melanin producing cell. Thyroxine cannot be converted to DOPA and dopaquinone when gene 1 or gene 2 is or are mutated. So melanin cannot be formed. Okay. So tyrosinase is an oxidase. That's why in the process of the reaction, you need oxygen there. Oxygen is needed to oxidize the substrate. So it's an oxidase enzyme that catalyzes oxidation of substrate. So it has 2 copper atoms in its active site which binds to the oxygen molecule. This one you don't have to know. This is just for your interest only. Okay, the tyrosinase enzyme from bacteria with L-DOPA in the active site, wherever. Okay. Now, so this is our skin, and right at the base here, you got this very huge cell with a lot of extension. This is the melanocyte. Okay, and tyrosinase is found in the membranes of the melanosome. So these are tiny little vesicles. We call them melanosome. So the tyrosinase is an enzyme that's found in the membrane of the melanosome in melanocyte. Okay. So these enzymes are needed to synthesize the melanin in the cell. Okay. So melanocyte is a melanin producing cell, which you can find it in the skin, in the iris of the eyes, in the hair follicles, means at the base of the hair where you've got cell there, etc. Okay, basically the skin, your eyes, and then your hair. Okay. So where is the location of melanocyte? If you look at our skin here, right, it's a top layer of the skin. Right at the bottom there, we've got the melanocyte with the extension. So they are the one that produce melanin for us. Okay. So this one is micrograph. Okay, to show you where are the melanocytes right at the base here. Okay, even the top also have got a little bit of color. Smooth muscle cell, you see here, huh? Okay, all these are smooth muscle cell. Right. By now you know how to identify the cell, but very easy to identify. Okay. This one looks more like elastic fiber, see? Same thickness throughout. So enzyme tyrosinase, interestingly, is also found in the apple and also potato, because you see when you cut the potato or the apple and leave it there in the air, right, after some time or very quickly they will turn brown, because tyrosinase, when it's exposed to the oxygen, it will actually catalyze the reaction to produce melanin. So that gives you the brown color, even in the fruit also, which is very interesting, right? Browning of fruits, browning of tubers like potato and fungi, okay, when they have been damaged and exposed to the air. So this one tells us that tyrosinase function in the presence of oxygen to oxidize the substrate into colored pigment. Okay, so second one is sickle cell anemia. So this is a revision because you've done it, right? Okay, sickle cell anemia is a phenotype and also a genetic disease due to the mutation in the allele, okay, in the gene HBB. HBB codes for beta chain of hemoglobin. Hemoglobin is made up of 4 polypeptide chains, right? 2 alpha, 2 beta. So the beta chain is coded by HBB gene. So where does the mutation occur? Mutation occurs in, this gene is found in chromosome number 6. Okay, and this mutation occurs in the 6th code, because it's a 6th amino acid change, it's a 6th code. Okay, in the DNA, in the 6th code, DNA is code, right? mRNA is codon. So in the 6th code, there is a mutation. Normal 6th code is CTT and CTC, but the mutated one is CAT and CAC. So there's a substitution, right? T is substituted by A, and then the triplet codon also will change in mRNA. Triplet code changes, triplet codon changes, and therefore the amino acid will also change. It becomes valine. So originally it's glutamic acid, right? This is glutamic acid, COOH, carboxylic acid. So it can form a negative charge and ionic bond. Now it's replaced with valine. Valine is actually hydrophobic R group. Cannot form ionic bond. So the original ionic bond will be broken. The entire structure of this beta chain of hemoglobin will change to become fibrous. Okay. So hemoglobin with two abnormal beta chain each will stick together to form long fiber when oxygen level is low. So this will happen when oxygen level in the body is low. The hemoglobin will start to stick together to form a long fibrous, long fiber in the red blood cell, pulling the red blood cell from biconcave dish shape to become sickle shape. So sickle stands for crescent. So you can see that this is normal biconcave dish shape and this normal hemoglobin. When there's lack of oxygen, the beta hemoglobin becomes fibrous and then they will join up together to form a very long fiber. So that will pull the shape of the hemoglobin of the red blood cell out to become crescent shape. Okay, so this is called sickle red blood cell or sickle cell. So this is micrograph of the blood to show you some sickle cell present in the blood. And sickle cell problem is that they are not so flexible. So when the blood flows to a smaller blood vessel, the sickle cell will block the blood vessel. So there will be no oxygen and nutrient supply to the targeted tissue. So the cell may die in targeted tissue. Okay. So then the person might also die depending on the severity of the tissue being damaged. Can be brain, can be heart, can be lung, can be anywhere, right? Kidney, liver. So therefore, a person who knows that he or she is born with sickle cell anemia, they must take care of themselves so that their body oxygen level will not go low. So they cannot go out and exercise like us. They cannot go to high altitude where the partial pressure oxygen is low. They have to take care. Okay, so if they take care, then they will survive, they'll be okay. Okay. So consequences of sickle cell anemia is fibrous hemoglobin cannot carry oxygen. So when the hemoglobin becomes fibrous, of course it cannot carry oxygen because it changed shape already. Okay, so therefore the red blood cell, the sickle cell will then be broken down and this person will be lack of normal red blood cell, so he'll be anemic. Okay, there'll be oxygen deficiency in the body. So this person will always be weak and a headache when you don't have enough oxygen in the brain, dizzy and frequently ill. Because body don't have enough oxygen, a lot of function cannot function properly. So quite weak, okay? Possible death also if too serious, right? Also can block capillaries and small arteries, reducing nutrients and oxygen to supply to the targeted tissue and damage organ may happen. Okay, so other features like enlarged lymph nodes, heart, joints, possible death. So this person must take care not to allow the body to not have enough oxygen. So that is a sickle cell anemia. So now we look at haemophilia. Okay, haemophilia. Haemophilia is a bleeding disease. Bleeding disease. It's a blood disease. The blood does not clot normally due to mutation in F8 gene. So the normal F8 gene code for factor 8, which is a blood clotting factor. This is needed for blood to clot. So when mutation occurs to F8 gene, it codes for defective factor 8. Factor 8 can be produced, but it's a different shape because different amino acid sequence. Okay, so therefore the blood cannot clot. So a hemophilia is a person with hemophilia. It is lack of blood clotting factor 8. So he experiences uncontrollable hemorrhage because when you go around, you might be hit on something, you may fall down, okay? And then you might bite your tongue or whatever. This will be injury. Usually injury will cause bleeding to happen. Sometimes internal bleeding happen, but the blood cannot clot. So the person may die due to internal bleeding also. Okay? So haemophilia is coded by a recessive sex-linked allele. So this is a recessive sex-linked disease. So XH is the normal allele which codes for factor A. So it is dominant. X small h is a recessive allele. This is the allele which codes for no factor VIII production, haemophilia. Okay. So haemophilia, the people with haemophilia is because the X capital H is mutated, so it becomes small h. Okay, haemophilia. So because haemophilia is sex-linked, so it's passed from father to daughter in the X chromosome. Father only got one X chromosome, will all be passed to the daughter, and mother will pass to the son because the son only need to receive one X chromosome with a small h, he will show haemophilia. Okay, so mother passed to son and father passed to daughter. Okay, so that is for haemophilia. So therefore, male is easier. There'll be higher chances for male to get sex-linked recessive disease than female. Correct? Because if for female to get haemophilia, the female must receive the X small h from father and X small h from mother. So the chances of both father and mother having X small h is actually very slim. Okay, but for male, as long as the mother is a carrier already can pass to the son, because the chances of the son getting the X small h is, hang on, so XH. So if the mother is heterozygous, chances of the son getting X small h is 50-50, right? So the chances of getting haemophilia is 50-50. But if the mother is homozygous, the chances of the son getting haemophilia is 100%. Because the X chromosome of the son comes from the mother. So if the mother is hemophilia, all the son will be hemophilia. But the daughter doesn't have to be hemophilia because the father can be normal. Then all the daughters are normal, but genotypically carrier. So if the father is normal, then all the daughter will be heterozygous. So daughter phenotypically is normal. Genotypically is a carrier. Okay, when you talk about carrier, you're talking about the genotype. Then because you're talking about the allele that's being carried by this individual. Carrier is XH, X capital H, X small h, because she's carrying the X small h causing the disease. So she's a carrier in terms of genotype. But phenotypically, she's normal. Okay? So do not write carrier for phenotype. Phenotype is normal. Okay, the last disease we're going to look at is Huntington's disease. This is a dominant disease. This is a dominant disease. So that means higher chances of getting and higher chances of showing phenotype. Okay. Huntington's disease is an inherited neurological disorder. That means neurological means it affects our nervous system. That means the central nervous system, brain, spinal cord, and also the peripheral nervous system. Okay, neurological. This is the meaning of neurological. Neuron, right? Neuron. Due to mutation to HTT gene. So this HTT gene code for the protein called huntingtin. So this is the only one that's protein, protein huntingtin. No, the rest of them are protein, but some of them are enzymes, some of them are beta chain of hemoglobin. Okay, this is a protein, not enzyme, which results in progressive mental brain deterioration, mental in terms of brain. The brain will deteriorate, okay, and involuntary movement means chorea will happen to this person because of deterioration of the brain. Chorea means dance-like movement. That means you cannot control movement of your muscles, right? So this person will like move maybe the hand or maybe the jaw, or maybe the eyes, and so on. Okay, so uncontrolled movement often associated with this Huntington's disease because the brain cells die. Atrophy means death of the cell. So the brain cells die and are lost. So the ventricles, means the fluid-filled cavity in the center of the brain, becomes larger and larger. So this is a symptom of this disease. The age of onset is variable, but most commonly in middle age. It means this person will not know he or she has the Huntington until the symptoms start to develop at a middle age. Okay, so this is the brain, and what they say is this ventricle. The ventricle part, which is fluid-filled, will be enlarged when the cells die. Okay, so left side is normal brain and right side is person with Huntington disease. Can you see the center here? The ventricle is actually enlarged because the brain cells around this region die. Okay. So Huntington's disease is because of mutation to the gene coding for huntingtin, and this mutation leads to a dominant allele. So the moment mutation occurs, then this person will definitely develop Huntington disease, dominant allele. Okay? So the mutation occurs in an unstable segment in the HTT gene in chromosome number 4, which code for protein huntingtin. Huntingtin. Okay, so this is the gene here, and this is a region where there's a repeated base sequence CAG, CAG, CAG. So because it's CAG, it codes for the same amino acid, right? Okay. So what happened in a person with Huntington is these repeated base sequences is actually longer than normal. They repeat more times than normal. Okay. So normally this sequence of DNA is repeated 10 to 35 times, but people with Huntington disease, this sequence here is repeated more than 35 times. So 36 to 120 times. So when it's repeated more than 35 times, then the number of amino acid here becomes more. So the entire huntingtin protein becomes longer. The polypeptide chain becomes longer. Okay, that leads to the atrophy, brain cell death. Okay, that's what has been found. Okay, so what are the chances of the offspring having Huntington? It depends on the parent's genotype, isn't it? Okay, so this is a parent with Huntington's disease and heterozygous. This is parent that's normal. Okay, homozygous recessive. So what is the chances of offspring getting the Huntington is actually 1 to 1, right? These two will get, these two will not get, so it's 1 to 1. Then for this parent is again, heterozygous dominant with Huntington's and heterozygous dominant with Huntington. So both parents also heterozygous. Okay, so therefore chances of offspring getting Huntington is actually 3 to 1. 3 to 1. Okay, so this is how you calculate the ratio. So that's why when we said that when two couples want to get married and have children, sometimes they have to go and do some genetic tests to find out whether they have got any genetic disease, and if they have, what is the chances of offspring getting the disease? So do they want to opt to have normal birth, normal pregnancy, or they want to do IVF? If they do IVF, they can actually use the genetic screening to screen the embryo. Okay, IVF, they're going to collect the sperm and egg and fuse all of them, and you get a lot of embryos. So you can actually screen the embryo and see whether it's defective and choose the one that's normal. That's one of the ways to choose, or else opt for adoption. I think in the past they opt for adoption when we cannot do genetic screening. Then you don't know which embryo is normal. So that's why it's important to do a genetic test if you want to have children. Okay, imagine that your first child has got Down syndrome. My friend's first child has got Down syndrome, and that time she's very young. So you can imagine that people say that you got 40 over years old, chances of getting Down syndrome kid is higher. But then my friend is only 20, almost 30 years old only. First child, Down syndrome. So better do screening, right? Okay. So what we have learned today is the effect of mutant allele on the phenotype. So when a normal gene is mutated to give a mutated allele, it can change the phenotype and give rise to genetic diseases. So genetic diseases is a type of phenotype. So you have looked at genetic diseases, which is albinism, sickle cell anemia, haemophilia, and Huntington disease. And they occur because of mutation to those genes, right, which code for certain type of protein, right? Thyroxine gene, which code for tyrosinase, needed to produce normal pigment melanin. HBB gene, which code for haemoglobin, which is a beta chain, okay, normal beta chain to carry oxygen. And F8 gene code for factor 8, which is a blood clotting factor needed for the blood to clot. HTT gene code for huntingtin protein. When it's normal, the brain is normal. When it's too long, because it's got repeated amino acids part is too long, then the person will have got Huntington's disease, where the brain cells will die. Okay, so that's all for today's lesson.